Hey there, everybody. This lecture is talking about enzymes. So we talked about how enzymes are a really special type of protein. These are a protein. And they're so special, in fact, that we have dedicated an entire talk to them. And in your textbook, you have an entire chapter to read about this, chapter 2.4. Make sure you go check that out, please. So we talked about how amino acids are the building blocks of proteins. So amino acids, these are the monomers of a protein. We want to go a little bit deeper into the structure of an amino acid. So amino acids have 4 components that you need to really know. They've got the central carbon, they've got an amino group, they've got a carboxyl group, and they have a variable R group. So the central carbon is sometimes also called the alpha carbon. The amino group is often represented as either, it could be an NH2 group, or sometimes if it's kind of on the other side, if the amino group is on the left-hand side of the amino acid drawing, then we put H2N. These are the same, these mean the same thing, right? A carboxyl group is also known as a carboxylic acid. And so that's why these are amino acids. And you saw this in fatty acids as well. They have a carboxylic acid group as well. And this needs to be changed here. Carboxylic acid. There we go. And just like the amino group, we can draw it in a few ways. We can either draw it like this, COOH, or if it's on this side, then we write COOH or HOOC. And sometimes you might also see it written out like this with its oxygens separated like that, or OH. And sometimes you might see it written out like this, COHO, like that. So these are all the same thing, right? These are all ways to write carboxylic acid. And then we've got something called a variable R group, and this is a side chain, and we'll talk more about that. But there are 20 different kinds of them, and they determine the R groups determine which amino acid it is. Okay, so these determine the amino acid. So here you can see the different parts. So we've got the central carbon here. Here's the central carbon. And then we've got over here, this is the amino group. You can see the amino group is here. And it's got a nitrogen and 2 hydrogens, right? So this is that NH2 group that we were talking about, right? This is the, and since it's on the left side here, it's on the left of the central carbon, this is called this would be the H2N group, right? Or the NH2 group, if it was on the other side. And then here we've got, this is the carboxylic acid group. Here. This is the carboxyl group, or the carboxylic acid group. And like I said, we can write this in a couple ways. We can write this COOH, or we could write this HOOC if it's on the other side, right? Here they've drawn the chemical bonds. And then down here, we've got the R group is down here. Here's the R group. And this depends on what, this will change depending on which amino acid it is. Right here, this is the central carbon is the other way I don't want to use that color. Let's use this color. This is the central carbon. You can just call it the central carbon. You don't have to call it the alpha carbon. Okay, so remember that that carbon can make 4 covalent bonds, so that's why it has 4 different parts to it, 4 different things coming off of it. All amino acids share this structure in common. They have a central carbon, they have an amino group on one side, they have a carboxyl group on the other side, and then they have the R group, which determines the amino acid. Okay, now amino acids are connected by these peptide bonds. We talked about that already. Okay, amino acids are connected with peptide bonds. And what's maybe a little bit extra here is that it's a carboxyl group attaching to an amino group. So the carboxyl group is attaching to the amino group, and that's what forms the peptide bond. All right, so there are 20 different types of amino acids, and that represents how many different R groups there are. So if there's 20 different amino acids, that means they each have a different R group. They can be really, really simple, like sometimes the R group is just a hydrogen, or it's just a CH3, a methyl group. This one here is getting a little more complicated. It's got some branches on it. Okay, this one has a ring. Here it's got a sulfur in it. But really, you don't need to memorize all of them. What you need to understand is that we can place them into groups. Okay, so there's 3 groups that you need to be able to place your amino acids into, either a polar group, a non-polar group, or a charged group. Okay, and I'll make it really obvious. If I give you a structure of an amino acid and I say, hey, look at this amino acid, what is it? Is it a polar amino acid, a non-polar, or a charged? You would be able to determine that pretty easily, okay? So polar R groups are R groups that contain electronegative atoms like oxygen or nitrogen. Don't forget sometimes sulfur, right? Sulfur. The other thing is that they're usually at the end of a side chain. Okay, so you can see here, these are the polar amino acids down here. And so you can see that the oxygen's at the end of the chain here. The oxygen is at the end here. The sulfur is at the end. The 2 oxygens here are at the end of the side chain. Here's got a nitrogen and an oxygen. These ones would be really polar. So they're pulling electrons towards them. So the distribution, electrons are not shared equally in these covalent bonds. The oxygens pull the electrons more towards them. And the nitrogen and the oxygen here, they will pull electrons through these chemical bonds, through these covalent bonds up towards them. So they'll have a slightly different charge there, right? Okay. Now, what else can we say? Sometimes they're in rings. So you can see here we've got a nitrogen in a ring. So this one here can also be a polar R group. But usually they're at the end of the side chain, at the end of a long chain. Okay, non-polar amino acids, they typically have hydrocarbon side chains, right? So they're usually these hydrocarbon side chains. Remember that a hydrocarbon is carbon and hydrogen only. Okay, so the more carbons and hydrogens you have, the more non-polar it is. And so you can see here, let's look. Carbon, carbon, carbon. So that's got 3 carbons and they all have their hydrogens on them, right? This one's got carbon and hydrogen here, right? Carbon and hydrogen here. This one here, carbon, 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 carbon. This is called a benzene ring. This is a this carbon ring here is very, very nonpolar, right? And then you've got a carbon here as well. So lots of carbons and hydrogens. Remember, all of these carbons also have hydrogens attached to them, right? They all have hydrogens on them. These ones here, hydrogen, hydrogen, hydrogen. So all of these carbons, we're just lazy. We don't need to write them all out. We just know that these ones have hydrogens on them, right? So these are very nonpolar. Here, this one here has a sulfur, but the sulfur is in the middle of the side chain. So that makes it nonpolar. I would probably not give you that one though, right? I would give you more like these ones here. Carbon, 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 carbon. Carbon, carbon, carbon. Carbon, 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 carbon. Lots of carbons and hydrogens, right? So you can see these are very, very nonpolar. Again, this one here, this one has an oxygen in it. I wouldn't give you this one. That's a cheeky question, but what you see is that you've got this, again, this ring with lots of carbons and hydrogens. It's very, very nonpolar. Okay, this one here, again, this one here has a nitrogen in it, even though it's nonpolar. Look at all the carbons and hydrogens. 1, 2, 3, 4, 5, 6, 7, 8 carbons. So that's very, very non-polar. Oh, sorry, 9 carbons. There's another one there. So that's a very non-polar group just because there are so many carbons and hydrogens there. Okay, but if you see them at the end, of the side chain of that carbon-hydrogen chain, then you've got, typically this is a more polar property, right? Electrons are going to get pulled towards these ends here. The electrons will get pulled towards these ends. Okay? Sorry, that's not a, oops. That's not a polar part there. So again, this one here, the oxygen's at the, kind of off to the side here, the electrons would get pulled this way. That might be a bit of a cheeky one. I probably wouldn't give you that one. Okay, I'll let biochemistry teach you all that stuff here. Right, and then there are positively and negatively charged side groups. Often this one, so what you'll see here is they'll usually have a charge. Right, so here's a positive charge. Here's, oh, sorry, that's the amino group. Don't count that one. Only look at the R groups. This one here has a negative charge. This one here has a negative charge. Now, whenever you see these nitrogens, these NH groups here, these ones here usually will have they'll usually have an extra hydrogen on them. When they have an extra hydrogen on them, that gives them a positive charge. So these ones often carry positive charges if they have an NH group on them. Just like this one over here, this one here is NH3 plus, right? So it's got an extra hydrogen, which gives it the positive charge because a hydrogen is just a proton. So they pick up protons is what they do. All right. But again, I would give you one with a charge on it. I would never give you like this one here. I would never give you this one here. I probably wouldn't give you. I would give you these ones here with the negative and positive charges clearly shown on them. All right, proteins have 4 levels of structure. So we've got the primary structure, which often we'll just give, we'll show it with like this 1' here. Secondary structure, which we usually give this 2 with a superscript 0. It's called 2'. This is 1'. Call this 1'. 2'. So primary structure, secondary structure, and we also have tertiary structure and quaternary structure. So the primary structure of a protein is just the polypeptide chain. It's just the polypeptide chain. Okay, so what it is, it's the order of the amino acids. So what's connecting them is the peptide bonds, okay, and the sequence of amino acids is the primary structure. Okay, I often think of this as like, this is one dimension. It's not really, but I consider it like 1D. It's not really, but close enough. The secondary structure is this here. So this is the secondary structure. Okay, and there's two there's two types. There's beta sheet and alpha helix, or just sheet and helix. And what's holding these two together is the hydrogen bonding between R groups and between the carboxyl and the amino groups. There can be hydrogen bonding. And so what you'll see if you look closely, here's the hydrogen bonds right here. Here's a hydrogen bond, hydrogen bond, hydrogen bond, hydrogen bond. Remember that this oxygen is going to be slightly negative, and this is a hydrogen here, right? This is an oxygen. This hydrogen is going to be slightly positive, and that's going to cause hydrogen bonding between them. Here we've got a hydrogen, here's an oxygen. That will be slightly negative oxygen, slightly positive hydrogen. That will create a hydrogen bond. So these are the H bonds here. H bonds. Okay? Now in beta sheet, typically what it is it's kind of this folded structure like this, whereas in helix is more of a twisting structure. Okay, so sheet, this is sheet, and it's typically more folded. You could say it's folded, or it's layered. And then over here, this is helix. And you could say that it's a spiral, or it's a coil, or it's twisting. Okay, you could say that it's winding, not winding, winding. They look the same. Okay, so that's secondary structure. Hydrogen bonds are holding these together. So if we look really closely here, if we get close, the hydrogen bonds would be forming between the layers like this. But with helix, it's usually, they're kind of skipping layers like this. So it's kind of like this. Okay, but the way that the hydrogen bonds form will either create sheet or helix. Okay, and that's secondary structure. Now, tertiary structure is essentially the three-dimensional shape. This is like the 3D shape of the protein. Okay, and this really gives the really the 3D shape of the protein provides, or no, determines the function of the protein. Okay, now what's causing the protein to fold into a three-dimensional shape is what's called hydrophobic interactions. Hydrophobic, in other words, non-polar interactions. So what you need to remember here is that polar R groups like to be with polar R groups, and non-polar, I'm just going to say NP, likes to be with non-polar R groups. Actually, no, let's write it all out for you. Let's make sure we write it all out. Non-polar with non-polar. Okay? And so you can see in this image here, we've got these non-polar regions. Here's a non-polar region, and here's a non-polar region, here's a non-polar region. Okay, so these here are non-polar amino acids. And they like to hang out together. They want to group together. And because they're hydrophobic, they tend to fold into the interior, right? So non-polar R groups, these nonpolar R groups. Let's do this. Nonpolar R groups, they tend to hide in the protein's interior, right? They want to be away from water because they're nonpolar. They are hydrophobic. They are fearful of the water. And that really determines, kind of determines the shape, right? Because they are trying to get away. The protein will create a shape where the nonpolar amino acids are touching water as little as possible. So that'll be the lowest energy shape, essentially. The polar R groups, though, they don't mind, right? Polar R groups, these ones here, they like water, right? They face watery environments. Okay, so that's like inside and outside the cell. Okay, so inside of the cell and outside of the cell has lots of water. So if it's inside the cell, then it'll just face outwards into that watery cellular environment. Okay, so that's why it doesn't really matter so much, because they like being out in the water, right? So they don't care about being in the water. It's these nonpolar groups that fold towards the inside of the protein to stay away from the water. Okay, and then the last structure is called quaternary structure. And quaternary structure is when you have more than one subunit. Okay, so protein, let's do this. So two or more protein subunits that come together to form a protein complex. So here in this example, this is hemoglobin. This is hemoglobin right here. And you can see we've got 1, 2, 3, 4 subunits, right? So this is a subunit. Let's do purple. Here's a subunit, right? This one here. This is a subunit. And then we've got this orange one here. This one here. This is another subunit. Let's do it black. There's another subunit. Right, and then this yellow one here, there's another subunit, and then this green one here, there's another subunit, right? So this is a subunit. This is another subunit. Okay, cool. Okay, and so together, they make the finished product, the finished protein, which is a quaternary protein. All right, so just to recap, the primary structure is the order of the amino acids in the polypeptide chain. Something to consider here is that, remember that the primary structure, this will determine the 3D shape, and the 3D shape determines the final function of the protein. So if you change, right, if you change the primary structure, then you change the shape, and you could alter the function. Okay, that's a really key concept to understand, is that if the primary structure determines the final function, right? Because it determines the 3D shape. But if you change the structure, then you change the shape and you alter the function. That can either be a good thing, a bad thing, or not really have any ultimate change. Secondary structure is the two-dimensional structure. It's the sheet or the helix, the layering or the twisting, right? Those are formed by hydrogen bonding. The tertiary structure is that three-dimensional structure. It's formed by the hydrophobic interactions. And then the quaternary structure is a protein complex, right? More than one protein subunit, right? More than one subunit, one protein subunit will make a protein complex. Okay, and then the last thing I'll say about folding is just that, yeah, again, the protein's final shape is determined by its primary structure, right? And yeah, again, just if we change the primary structure, then you change the shape, which ultimately will change the function. Okay, that folding occurs spontaneously, and it happens due to the hydrogen bonding that occurs in the secondary structure, the nonpolar interactions that happen to make the tertiary structure. But then there's also other things like disulfide bridges. These ones here, here's a disulfide bridge. This is a covalent bond, right? And we talked about how there's positively charged and negatively charged R groups, and these can form ionic bonds, right? Ionic bonds and covalent bonds are quite strong. They're stronger than hydrogen bonds and nonpolar interactions. So there's all sorts of different interactions that help with the folding of these proteins, yeah. And so if you change one of these amino acids, essentially you could ruin an ionic bond, or you could destroy, like if you change this amino acid, you could destroy this disulfide bridge. Or if you put here a nonpolar amino acid, well, now hydrogen bonding doesn't occur. Or if you put a polar amino acid here, or three polar amino acids together, then this nonpolar interaction doesn't occur. So if we change the primary structure, we might change the shape and we might change the function. That's a really important concept. All right, we'll continue in the next video with denaturation and with enzymes, structure and function.